Hello friends, today we'll discuss about immunity, starting with the definition. It is the ability of body to resist the pathological agents like bacteria, viruses, toxic substances, etc. Immunity is mainly of two types. First, that is innate immunity, which is also known as non-specific immunity or natural immunity. And second variety is acquired or specific immunity, which is also known as artificial or adaptive immunity. Now, let us first discuss the difference between innate and acquired immunity. Innate or natural, that is available since birth. Acquired immunity, that is acquired after birth, and that is after exposure to the microorganism. Innate immunity, other name is non-specific, that is non-specific to particular microorganism, whereas acquired immunity is also known as specific immunity, which is specific for each species of the microorganism, and it also has immunological memory, means on second or subsequent exposure to the same antigen produces more rapid and greater response. Another difference between natural and acquired immunity is natural immunity is able to recognize and respond immediately, whereas acquired immunity it requires time to be fully developed. Now, let us start with innate or non-specific or natural immunity. As we have discussed, it is present since birth. It is inborn capacity of body to provide resistance to the pathogens and their toxic products. It is non-specific and it is due to genetic and constitutional makeup of an individual. Innate immunity eliminates the organism and therefore it prevents development of disease. It is also known as first line of defense. Some of the examples of innate immunity, or you can say mechanisms involved. First and foremost, that is mechanical barriers like skin and mucosa of our body. Second, that is surface secretions. For example, secretions of our sebaceous gland, which contains saturated and unsaturated fatty acids, which kills the microorganism. Saliva has inhibitory effect on the microorganism. Gastric juice, which contains enzymes, as well as HCl, which is acidic in nature and therefore it hydrolyzes the microorganism. Tears, they wash away the particles as well as they also contain hydrolytic enzyme which is known as lysozyme and it destroys the microorganism. Some examples of humoral defense mechanism. Humoral means this defense mechanisms they pass through humus, means body fluids. First and foremost is lysozyme. This is the enzyme which is present in various secretions like nasal secretion, salivary secretions and tear, which is mucolytic enzyme, and it kills the microorganism. The mechanism is it splits the sugar of structural mucopeptides which is present in the cell wall of the microorganism. Second is certain basic polypeptides are there. They also have non-specific microorganism destruction activity which is known as microbicidal activity, and it also inactivates the gram-positive bacteria. Another example of humoral defense mechanism is complement system. This complement proteins, they have lytic and several other defense effects on the foreign substances. Another example is interferon. These are antiviral substances which are produced by various cells. Cellular defense mechanisms, which includes phagocytosis, which is the property of neutrophils, monocytes and macrophages. Second cellular defense mechanism is natural killer cells. These cells are non-T, non-B type of lymphocytes and they destroy foreign cells. Also they destroy tumor cells and infected cells. Cellular defense mechanism also includes eosinophilic granules which contains enzymes as well as toxic molecules. And these enzymes and toxic molecules they act against the larva of the helminths. Next is acquired immunity. As we have discussed, that is acquired after birth. It is the resistance against specific antigen. It is more powerful than innate immunity and important component of acquired immunity is lymphocytes. According to the lymphocyte, the type of acquired immunity is cellular immunity, which is by T cells, and humoral immunity, which is by B cells. Also, it is divided in active and passive acquired immunity. Let us first discuss active and passive acquired immunity. Active acquired immunity. This is acquired by synthesis of antibodies as well as production of immunocompetent cells. This active immunity is further divided in natural active immunity. For example, if we have any infection, either it is clinical or subclinical infection, it activates our immune mechanism. So this is the example of natural active immunity. Artificial active immunity, that is artificially acquired by vaccination. When we are giving vaccination, at that time we are stimulating our immune mechanism to produce antibodies, and this is primary response. So actually whenever there is infection, at that time our body produces large amount of antibodies. Next is passive immunity. Here these antibodies are transferred in ready-made form, which is again divided in natural and artificial passive immunity. Natural passive immunity, that is transfer of antibodies, that is from mother to the fetus. For example, through placenta, IgG type of antibodies are transferred, as well as through breast milk, IgA antibodies are transferred. And artificial passive immunity, this is by injecting ready-made form of antibodies in immunocompromised individual, like serum containing antibodies. They are injected when the immunity of the individual is suppressed. Now, difference between active and passive immunity. In active immunity, antigen is introduced and antibodies are produced, whereas in passive immunity, we are directly injecting antibodies, either naturally or artificially. In active immunity, there is negative phase, means amount of antibody production is less initially, whereas this kind of negative phase is absent in passive immunity. In active immunity, there is latent period. Latent period is the time interval between the infection and production of antibody, whereas this latent period is absent in passive immunity. In active immunity, secondary response is enhanced. Why? Because there is immunological memory. First time antibody production also produces memory cells. They increase the production of antibodies on second and subsequent exposure, whereas this kind of immunological memory is absent in passive immunity. Duration of active immunity is long lasting, whereas for passive immunity it is short lasting. Active immunity is more effective and it provides better protection, whereas passive immunity is less effective and it provides less protection. Active immunity is not applicable in the immunodeficient individual, whereas passive immunity is given in the immunodeficient individual. Now, we discuss about antigens. Definition: antigens are the substances. They induce specific immune reactions in our body. Structurally, they are proteins. Some of them are carbohydrates, lipids, and nucleic acid. Antigens, they have property of specificity, and it is due to specific areas present on the antigen. These areas are known as determinant sites or epitopes. Next is histocompatibility antigens. These are the antigens present on the plasma membrane of cell of each individual of a species. These antigens, they are encoded by the genes which are known as histocompatibility genes, and they form MHC, major histocompatibility complex. These genes are located on the short arm of chromosome number 6. Another terminology is HLA, human leukocyte antigen. The MHC, major histocompatibility complex present on the surface of leukocyte, is known as HLA. No two persons except identical twins, they have same MHC proteins. There are mainly three subclasses of MHC gene: MHC1, MHC2 and MHC3. Now, what is MHC and HLA system? MHC, that is a group of molecules present on the short arm of the chromosome number 6, and HLA antigen system, it is small groups of genes present in MHC molecules. Two types of MHC molecules are important for we people. Type 1, that is present on all the cells. This type 1 MHC molecules, they present antigen to the cytotoxic T cells. Second variety is type 2. This type 2 MHC molecules are present on B cells and macrophages, and this MHC2 presents antigen to helper T cells. Now, this antigens are of again two types: autoantigens, which are also known as self antigens, example is blood group antigens, and foreign antigens, which are known as non-self antigens, and they are present as a receptors on the cell membrane, also may be toxins, tissue transplants and allergen. They all act as foreign or non-self antigen. Now, these non-self antigens are again classified in two varieties. Number 1, the antigens which produces development of immunity, or you can say they produce antibodies. And second variety, the antigens which produce allergic reaction. Next is antibodies. Antibodies are proteins which are produced by plasma cells, and they are produced in response to entry of antigen. Antibodies are immunoglobulins, and they have 4 polypeptide chains, 2 heavy chains and 2 light chains. They are linked by disulfide linkages. These immunoglobulins, they are mainly gamma globulins, which are 20% of total plasma proteins, and they are of 5 varieties: IgA, IgG, IgM, IgD and IgE. Now, structure of antibody. Molecular weight of antibody, that is from 1,50,000 to 9 lakh. Antibody is Y-shaped molecule made up of 4 polypeptide chains. There are 2 heavy chains and 2 light chains. Heavy chains having 440 amino acids and light chains having 220 amino acids, and they are held together by disulfide linkages, you can see here. They have mainly two regions: constant region and variable region. Now, constant region, here it is the region having same amino acid, also known as FC region. This is the part where complement fixes. It is known as complement fixation site. And second is variable region, which has different number as well as sequence of amino acids, and it is known as antigen binding site, also known as FAB region. Now, we'll discuss about heavy chains. Heavy chains, they have molecular weight double than light chains. Structurally and autogenetically, they are distinct for each class of immunoglobulin. For example, if the immunoglobulin is IgA, it has alpha chain, IgD delta chain, IgE epsilon chain, IgG gamma chains and IgM mu chain, but it is identical for given molecule of immunoglobulin. In heavy chains, there are 4 loops of 110 amino acids. It has variable as well as constant region. Variable is known as VH, and constant regions, they are CH1, CH2 and CH3. Light chains, the molecular weight is 25,000. It is of two types, kappa and lambda chain. Kappa and lambda chain occurs in a ratio of about 2:1. Similar to heavy chain, variable region of L chain, light chain, is designated as VL. and constant region of light chain is designated as Cl, and it contains two loops, each containing 110 amino acid. Each loop forms a globular domain. Now we discuss about various immunoglobulins and their functions. IgG, it is 71% of total immunoglobulins. Normal level, that is 0.8 to 1.6 gram per deciliter. Molecular weight is about 1,60,000. Heavy chain, that is gamma chain. Light chains are kappa and lambda chains. Functions, these are the antibodies of secondary immune response. They are distributed equally between blood and tissue fluid. They can cross the placenta from mother to fetus, and that is by active transport. Example is antibodies of Rh blood group system are of IgG variety. They can also serve as opsonine and they promote chemostatic activity of white blood cell. IgA, 22% of total immunoglobulin, 140 to 420 mg per deciliter. Molecular weight is 1,70,000. They have alpha chain as a heavy chain and kappa and lambda light chains. Functions, they are present in the secretions, present in the plasma, tears, saliva, intestinal juice, respiratory secretions and colostrum. Unlike IgG, they cannot cross the placenta. They lyse the bacteria in presence of lysozyme, which occurs in the secretion containing IgA, and therefore they protect mucous surfaces and they provide localized protection. Next is IgM, 7% of total immunoglobulin, 50 to 200 mg per deciliter. Molecular weight is 1 lakh. Heavy chain is mu chain and light chains are kappa and lambda chains. Functions, they produce primary immune response, as well as they also activate complement system, they promote the phagocytosis and they cause cell lysis by digesting the holes in the cell membrane at the site of antibody attachment. These are more effective than IgG antibodies in lysing the cell. Next, IgE, present in very small amount, 0.03 mg per deciliter. Molecular weight 1,85,000. Heavy chain is epsilon chain. Light chains are kappa and lambda chains. Functions, these immunoglobulins are heat labile and skin sensitizing antibodies, which are known as reagins, and they are responsible for immediate hypersensitivity reactions. Next variety is IgD, also present in very small amount, 3 mg per deciliter. Molecular weight is 1,50,000. They have delta chain as heavy chain and kappa and lambda chains as light chains. Functions, they are present on the surface of B lymphocytes with IgM and they are involved in antigen recognition. So this is all about general introduction of immunity as well as antigen and antibodies. Thank you.